Hi there. Every drug that enters the body faces a critical challenge. Before it can reach the bloodstream, before it can reach its site of action, it must first cross one fundamental structure: the plasma membrane. The plasma membrane is not just a covering around the cell. It is a highly selective, dynamic barrier that controls what enters and leaves the cell. According to the fluid mosaic model, the membrane is not rigid. It is dynamic, constantly moving, composed of lipids and proteins arranged like a mosaic floating in a fluid sea. Its core structure is the lipid bilayer, mainly made of phospholipids, cholesterol, glycolipids, and membrane proteins. Let's start with the key building blocks. Phospholipids. Each phospholipid is composed of a glycerol backbone, two fatty acid tails, one saturated and one unsaturated, and a phosphate-linked head group. What makes them special? They're amphipathic. They have both a water-loving, hydrophilic head and water-fearing, that is, hydrophobic tails. In water, these molecules form a bilayer called a phospholipid bilayer. The hydrophilic heads face the watery environments inside and outside the cell, while the hydrophobic tails point inward, away from water. This forms the basic fabric of the plasma membrane. Because of their amphipathic nature, phospholipids can spontaneously form bilayers, micelles, or even liposomes, tiny bubbles, depending on their tail structure. The head contains a negatively charged phosphate group and a variable R group, making it interact well with water. The tails, on the other hand, are nonpolar and form a barrier to polar or charged substances. Cholesterol molecules float within the membrane. With their 4 fused carbon rings, they help modulate the membrane's fluidity, keeping it flexible but stable. If the membrane becomes too rigid, cholesterol makes it more flexible. If it gets too fluid, cholesterol helps stabilize it. Carbohydrate groups are only found on the membrane's outer surface. When attached to proteins, they form glycoproteins. When attached to lipids, they form glycolipids. These play a big role in cell recognition and signaling. Glycolipids, located mainly on the outer surface, contribute to membrane asymmetry and cellular identity. Together with carbohydrate chains attached to lipids and proteins, they form the glycocalyx, a carbohydrate-rich outer layer responsible for cell recognition, adhesion, and protection. By mass, the typical human cell membrane is about 50% protein, 40% lipid, and 10% carbohydrate. Membrane proteins are another key component. Integral membrane proteins act as anchors, embedding in the cell membrane due to their hydrophobic regions. Proteins that penetrate only partway into the membrane are referred to as monotopic proteins. In contrast, transmembrane proteins span the entire membrane, connecting the cell's interior and exterior. An example of a monotopic protein is cytochrome B5. The buried sections are hydrophobic, and the exposed parts are hydrophilic, so they interact with both the inner and outer environments. Their structure often includes spiral-shaped alpha helices or barrel-shaped beta sheets. Proteins may cross the membrane once or traverse it up to 12 times, often forming channels for ions or small molecules. Now, let's break down the classification of transmembrane proteins, types 1 to 4, based on their orientation and how many times they cross the lipid bilayer. These are all integral proteins, meaning they are permanently anchored within the membrane. Not all membrane proteins are inserted the same way. First up is type 1. This is a single-pass protein, meaning it crosses the membrane only once. The defining feature here is that the N-terminus, the start of the protein, stays on the extracellular side, while the C-terminus faces the cytoplasm. These often act as receptors for outside signals. For example, insulin receptor. Type 2 is also a single-pass protein, but it's essentially the mirror image of type 1. Here, the C-terminus is on the outside, and the N-terminus remains inside the cell. A classic example of this is the transferrin receptor, which helps the cell take up iron. Now we move to type 3, which is a multipass protein. This is a single polypeptide chain that weaves back and forth through the membrane multiple times. Because it's one continuous string, it can form complex shapes, like the light-sensitive rhodopsin in your eyes. For example, cytochrome P450. Finally, type 4 involves multiple separate polypeptide chains that come together to form a single functional unit, like a channel or a pore. Think of this like several independent type 1 or type 2 proteins grouping up to create a tunnel for ions or water to pass through. For example, G-protein coupled receptors. Next, we'll learn about peripheral or extrinsic proteins, the specialists of the cell membrane. Peripheral proteins temporarily attach to the inner or outer surfaces of the membrane, unlike integral proteins that penetrate the membrane's oily core. They usually connect to either integral proteins or to the polar heads of phospholipids. Peripheral proteins are mainly hydrophilic, meaning they love water. They often interact with the watery environment inside or outside the cell and with the polar heads of the lipids. Structurally, they have alpha helices and beta sheets, but lack the long stretches of oily amino acids that let integral proteins bury themselves in the membrane. Some have special binding domains, such as pH domains, that help them bind to specific lipids. Peripheral proteins attach to the membrane through weak, non-covalent interactions, hydrogen bonds with polar lipid heads, electrostatic interactions with charged protein regions, and sometimes small hydrophobic loops just dip into the surface. Because these bonds are weak, you can remove peripheral proteins with mild treatments, like changing pH or adding salt. No need to break up the membrane. There are 3 main types, based on where and how they attach. First is cytosolic, inside the cell, often involved in signaling or supporting the cytoskeleton. For example, spectrin and actin help give red blood cells their shape. The second is extracellular, outside the cell, involved in communication or adhesion. One example is acetylcholinesterase, which resets muscle contraction. And the third is transient. These proteins attach only when they receive a signal, like protein kinase C, which moves to the membrane to start a signaling chain and then detaches. Some key peripheral proteins include cytochrome C, vital for energy production in mitochondria, and protein kinase C, important for signal transduction. Let's look into the 6 primary functions of these membrane proteins. First, we have transport. Since the lipid bilayer is hydrophobic, polar molecules can't just walk through. Ion channels act like open tunnels for specific ions, like sodium. Glucose carriers change shape to ferry larger molecules across. And ion pumps, like the sodium-potassium pump, use ATP energy to move molecules against their concentration gradient, essentially acting as the cell's biological bilge pump. Next, notice the proteins with carbohydrate chains attached. These are glycoproteins. They handle cell recognition, acting like ID tags, so your immune system knows which cells belong to you. Right next to them are adhesion proteins, which act like molecular Velcro, hooking cells together to form tissues. Next, we see signal transduction. These receptor proteins have a specific binding site for external signaling molecules, like hormones. When a molecule binds, the protein triggers a chemical relay race inside the cell, changing the cell's behavior without the signaling molecule ever having to enter. Some proteins are actually enzymes. Instead of moving things, they catalyze chemical reactions right at the membrane surface. You'll often see these grouped together as a team to carry out sequential steps of a metabolic pathway. Finally, we have the anchors. These proteins attach to the cytoskeleton on the inside and the extracellular matrix on the outside. They maintain the cell's shape and help fix the location of other membrane proteins so they don't just drift away. So, the plasma membrane is not just a barrier. It is a controlled gateway system. Why does this matter for drug absorption? Because the membrane is selectively permeable. Lipid-soluble substances can diffuse directly through the bilayer. Polar or charged molecules require specialized membrane proteins. This selective nature ensures that the cell absorbs essential nutrients, necessary ions, and important molecules, while blocking harmful substances. The plasma membrane doesn't just define the cell's boundary, it controls survival through regulated absorption. Now let's move from structure to function. When a drug crosses an epithelial barrier, such as the intestinal lining, it can take two anatomical pathways: paracellular route, between cells, and two, transcellular route, through cells. These are anatomical routes, not transport mechanisms like active transport or diffusion. First is the paracellular route, between cells. In this pathway, the drug passes through small spaces between adjacent epithelial cells. These spaces are sealed by tight junctions. Only very small, hydrophilic molecules can pass here. Examples include small ions, urea, and some small water-soluble drugs. The drug remains in the aqueous phase and never actually enters the cell. This route becomes more significant in neonates and during inflammation, in diseases where tight junctions are loosened. However, under normal conditions, tight junctions are extremely narrow, limiting absorption through this pathway. Now we come to the main pathway, the transcellular route, through cells. This is the primary route for most drugs. Here, the drug crosses the apical membrane, passes through the cytoplasm, and exits via the basolateral membrane. Because the membrane interior is lipid-rich and hydrophobic, lipid-soluble, non-ionized drugs cross easily. That's why most orally administered drugs are absorbed via this route. Examples include diazepam and aspirin. So, to summarize, the plasma membrane's lipid bilayer creates a selective barrier. Most drugs cross epithelial barriers through cells, the transcellular route, and only a minority of small hydrophilic molecules pass between cells via the paracellular route. The structure of the membrane determines the pathway, and the pathway determines absorption. So today, we understood the structure of the plasma membrane, not just as a protective covering, but as a highly selective and dynamic barrier that controls absorption. Now the next important question is, how do substances actually cross this membrane? In our next video, we'll explore the transport mechanisms of paracellular and transcellular routes and the factors that affect absorption. I'll see you in the next video. Thanks for watching, and don't forget to like, subscribe, and hit the bell to stay updated with our pharmacology series.